Hello, everyone, and welcome back to Bio 181. In this lecture, we will be discussing Mendel's experiments in heredity. In this chapter, we'll discuss genetic and hereditary terms, the principle of segregation and independent assortment, and using Punnett squares to solve for genotypes and phenotypes. For thousands of years, humans have depended on their working understanding of inheritance. And we have used that in order to breed animals that were better suited to our needs. Here, the mouflon is thought to most closely resemble the ancestor of modern domestic sheep. And since then, sheep have come a long way. Shrek was a sheep that had left his herd for 6 years, at which point it's obvious to see that Shrek was not going to be successful all on his own. We can now see how sheep have adapted quite well to take advantage of a shared relationship with humans to procreate to populations high in number, but are not well suited as the mouflon to exist independently of us. This process was not merely a consequence of coevolution, but of humans' understanding of inheritance in order to breed specific traits. In this lecture, there are terms we commonly use in our day-to-day lives, but we must be careful to understand their specific meanings in this chapter. Inheritance is the specific transmission of qualities from parent to offspring, and this comes from the observation that the traits of offspring tend to resemble the traits of their parents. A trait is an inherited characteristic of an organism, either physical or behavioral. The key being an inherited trait is that it's inherited. If it isn't inherited, like hair color, then it isn't a trait. For example, an injury or memory would not be examples of traits. Take a moment, think of some of your own human traits. The generative fluid hypothesis states that offspring receive a blend of their parents' samples of, quote, generative fluid and end up with intermediate traits. This would be like mixing watercolors blue and red together to get a color purple. You wouldn't get red or blue, but something in between. However, this is not what is actually observed in nature. When experiments are conducted on a population of flowering plants, we can see that some of their offspring, called generation 2, are red, some are pink, and some are white. So we clearly need to revise this hypothesis. Gregor Mendel was a monk from the 1800s Czech Republic, which at the time was part of the Austrian Empire, who studied the process of the inheritance of traits of pea plants and is referred to as the father of genetics for his groundbreaking work. Mendel studied inheritance of traits long before we had any idea what DNA was. Here is a drawing of Mendel's using a brush to pollinate specific pea flowers. By taking pollen from one flower and brushing it into another, he is simulating what a bee would do in a controlled manner. Pea plants were useful because they are easy to grow and quick to grow, and they have many varieties with different traits to examine, and have been studied extensively by other researchers. So there is a basic scientific understanding of their nature already. Pea flowers contain both male and female parts in the same flower. However, the male parts can be removed so that flowers don't self-fertilize, or vice versa, the female parts. And then different flowers can be mated manually using Mendel's technique. Here's an illustration of a pea flower. Pea flowers have both male and female parts in the same flower. We can see the male portion as the stamen, the anther contains the male gametes that we refer to as pollen. That pollen can then be delivered to the carpel, which is the female portion. The carpel is made up of the stigma, style, and ovary. The ovary is the location that harbors the female gametes. And here we can see how a brush can be used to collect pollen grains in order to be applied to a specific female flower. And in this case, the source of the pollen is from a red flower, and the destination female flower is white. In Mendel's experiments, he was able to create true breeding varieties. A true breeding organism is one that always passes down certain traits when bred with the same true breeding organisms to their offspring. Mendel would allow one variety of pea to self cross for multiple generations to ensure that it consistently produced offspring of the same variety. We can follow one of Mendel's experiments between true breeding pea plants with round seeds, which basically means a smooth texture. These seeds were crossed with other true breeding female originating from wrinkled seeds. So we have true breeding male round seeds and true breeding female wrinkled seeds. To cross simply means to breed, as was shown in the pollination slides. The offspring in the first generation are referred to as the F1 generation. If you are interested, F stands for filial, meaning the offspring. Something interesting occurred with this offspring, and that was that all seeds produced were round seeds. And this experiment did not support the hypothesis of generative fluid. Just as interesting, when the F1 seeds were planted and crossed with each other, an unusual ratio of round to wrinkled seeds were generated in the F2 generation. You can find the ratio by taking the number of round seeds and dividing them by the number of wrinkled seeds. Again, if the generative fluid hypothesis were correct, we would expect to see F1 seeds to be somewhat smooth, but not as wrinkly as the wrinkled parents. And we certainly wouldn't expect the second generation to have seeds wrinkly like the female parents in the beginning and smooth like the male parents in the beginning. So what is going on here? Checkpoint 1. What ratio of round peas to wrinkled peas did Mendel get in his F2 generation data? Mendel repeated this experiment using true breeding yellow peas and true breeding green peas. And again, the F1 generation produced 100% yellow peas. And the second generation produced 6,022 peas, yellow peas, and 2,001 green peas. This ratio would be roughly 3 to 1. So Mendel was able to confirm this pattern of inheritance between true breeding peas in both F1 and F2 generations for both round and wrinkled seeds, as well as yellow and green peas. So, how can we explain this phenomenon? Mendel confirmed these patterns of inheritance in a variety of other traits as well. If we look on the far right column, all of the parental traits were found in the F2 plants to be in a ratio of roughly 3 to 1. Well, Mendel came up with a really fascinating hypothesis to try and describe this. Mendel's first hypothesis or principle addressed the following question: why are the F2 ratios always close to 3 to 1? It was clear to Mendel that inheritance was not a substance that was just mixed together from parents, as the generative fluid hypothesis claimed. He proposed a principle of segregation instead that stated, in any given plant, each trait is determined by precisely two of these particles, one from the male and one from the female. Of a, and this collection of particles contained one particle that was dominant over the other. In other words, one of these particles, if present, would rule what sort of expression or outwardly behavior or physical characteristic that organism had. It was only about 100 years later that the particles Mendel was referring to turned out to be genes. Genes are made up of specific sequences of DNA located in specific positions on chromosomes, and that gametes only have one of the parent's two chromosomes for any given gene. In the image, we can see how a single set of genes is isolated through the process of meiosis, leading to haploid cells called gametes. If the primary sex cell, that one at the top, has both a dominating trait on one of its homologous chromosomes and a recessive trait on the other, then after meiosis 1 and meiosis 2, there will be 4 cells, 2 with the dominant gene and 2 with the recessive gene. But more on this later. Checkpoint 2. The genetic material is DNA, which is composed of nucleotides. Describe the structure of a DNA nucleotide. Include in your discussion the 3 main components of the DNA nucleotide and how are nucleotides attached to one another. Okay, DNA and genes. There are 4 different nitrogenous bases: adenine, guanine, thymine, and cytosine. The order of the nucleotides in a molecule of DNA is a language spelling out instructions for cellular processes. Additionally, DNA is double-stranded, so one strand is associated with another strand or polymer, as shown in the picture. Genes can have variations in them, just like how there may be different recipes for chocolate cookies. These variations are called alleles. Alleles are any of the alternative forms of a gene that vary in the order and content of their nucleotides. As an example, we can see that a change in the genetic sequence of this gene, containing cytosine, guanine, and adenine in red, can lead to the trait of freckles on an individual's skin, while if in the same place thymine, and guanine exist, then there are no freckles. So that would be the first allele and the second allele of that gene. This brings us to an essential set of terms we need to be aware of. This is genotype and phenotype. Genotype describes what genetic alleles an individual possesses. So when we discuss genotype, we mean to say what the genes look like, not the individual. Phenotype is not the observable properties of a trait. Phenotype is the observable properties of a trait or set of traits. So when we discuss phenotype, we mean to say what the individual's traits are. As an example, Rudolph has the allele for freckles on his maternal chromosome and the allele for no freckles on his paternal chromosome. That is his genotype. Look at Rudolph. We can see his phenotype, and his phenotype is that of one having the traits of freckles. Confused yet? Don't be. See, just because his phenotype is that of being freckles doesn't tell us exactly what's going on with his alleles or his genotype. It could be possible that he has the allele for freckles on both chromosomes, or just the maternal or paternal chromosome. And this is one of the reasons why it's important to know the difference between phenotype and genotype. So, part of Mendel's principle of segregation was that one trait was dominant over the other. The other trait is referred to as recessive. A dominant allele is one that overrides the expression of another allele when present. It is indicated by a capital letter. A recessive allele is one whose expression gets overridden by another allele when present. It is indicated by a lowercase letter. In our previous example, Rudolph had one allele for freckles and the other allele without freckles. However, Rudolph exhibited freckles. In humans, the allele for presence of freckles is dominant. An individual who has one of each allele will have freckles. For a trait with two alleles in a diploid organism, there are four possible genotypes. And remember that in genetics, the capital letter represents the dominant and the lowercase represents recessive. Capital F means that the allele has the DNA sequence for freckles, because remember, freckles are dominant. And lowercase f means that the allele does not have the sequence for freckles. And there are four different possibilities: capital F, capital F, capital F, lowercase f for the maternal side, and capital F for the paternal side, or two lowercase f's. We can see that anytime there's at least one capital F, the individual will have freckles, but without the freckle allele, capital F, to recessive lowercase f's, that individual will not have freckles. So the phenotype is the same for the first three, even though those first three have different genotypes. Now, let's define the term homozygous, which refers to an individual or locus with identical copies of an allele for a given gene or on the homologous chromosome. So let's look at an example here. In this case, having both alleles for freckles, two capital F's, or both alleles without the genetic sequence for freckles, two lowercase f's, these two individuals are homozygous. This might help a little bit more. Understanding heterozygous, which is the opposite. This refers to an individual or locus on the chromosome with different alleles for a given gene on homologous chromosomes. So either the maternal or the paternal has the dominant allele, and the other homologous chromosome in the set has the recessive allele. So it could be capital F, lowercase f, or lowercase f, capital F. Those are heterozygous. Homo means same, hetero means other. Mendel studied traits that were ruled by only a pair of alleles, one on each chromosome. One allele was dominant over the other one, so smooth texture was dominant over wrinkled texture. And yellow skin was dominant over green skin, etc. Checkpoint 3. Use the letter capital T/lowercase t to indicate the different possible genotypes that a pea plant could have for the gene governing seed texture. Remember, we're doing genotypes here. And their corresponding phenotypes. Remember, smooth texture is dominant. Specify in each example which allele is on the maternal or paternal chromosome. Now, Mendel was very smart in the way he set up the experiments. He was sure to start with true breeding strains. Again, this means that if you breed a true breeding parent with another true breeding parent of the same breed, they both have the same allele, then the offspring will have the same allele and they will have the same phenotypes. So he took two true breeding strains for their traits, one being round or smooth and the other being wrinkled. And then he crossed them with the other true breeding strain. Again, a true breeding strain for round or wrinkled seeds has a genotype for those alleles that is homozygous. Two capital T's for a round true breeding seed and two lowercase t's for a wrinkled one. Let's examine the gametes of these homozygous parents, and this will help us assess the F1 generation. So if we take these gametes from the round seeds and the wrinkled seeds, which means that they, instead of having two copies of the one pair of homologous chromosomes, now they just have a single because they're haploid, recall. So since they are homozygous, 100% of the male gametes contain a chromosome with the allele capital T. 100% of the female gametes contain a chromosome with the allele lowercase t. And when the two parents mate, offspring will get one copy of each, the result of which will be a heterozygous offspring with a genotype of one round and one wrinkled allele. And this explains why 100% of the F1 generation is round. The round, or capital T, allele is dominant. So all the plant needs is one of these alleles in its homologous set here, and it will determine the expression, just like an allele that is dominant for freckles in humans will. Take the F1 generation now, and let's look at their gametes. Both the female and male parts of the flower, ovum, and pollen are heterozygous. As mentioned in the previous slide, because of this, the female gametes can carry the round or wrinkled allele. And the same is true for the male gametes. So what happens when we brush the pollen of an F1 plant onto the stigma of another F1 plant? Well, let's go through the different possibilities. There are four equally possible scenarios. Scenario 1 is that a round allele from the male joins a round allele from the female. The resulting fertilized embryo has a genotype now of capital T, and the phenotype of which will be round seeds. Scenario 2 is that a round allele from the male joins a wrinkled allele from a female. The resulting fertilized embryo is now capital T, lowercase t. The phenotype of this plant will also have round seeds because the dominant round seed allele will determine the physical traits. Scenario 3 is that a wrinkled allele from the male joins a round allele from the female. The resulting fertilized embryo is now lowercase t and a capital case T. The phenotype of this plant will also have round seeds. We can see we're getting a lot of round seeds here. Scenario 4 is that a wrinkled allele from the male joins a wrinkled allele from the female. The resulting fertilized embryo is now lowercase t and lowercase t. Unlike the other three scenarios, the phenotype resulting from this genotype will be wrinkled. And that's what happens in the F2 generation in Mendel's experiment. What phenotype for P texture would each of the following offspring exhibit? Remember that smooth texture is dominant to wrinkled texture. Okay, Mendel's first hypothesis was the principle of segregation. It was able to explain why 100% of the F1 generation plants were round textured, and why the F2 generation exhibited a 3-to-1 round to wrinkled texture ratio. Mendel's principle of segregation, the Punnett square. In the previous slides, we went through the mechanisms of gamete production, the different scenarios, and fertilization to understand the phenotypes and ratios of the F1 generation and the F2 generations. The Punnett square is a tool for calculating these offspring ratios to do the following steps. So the first step we need to do this, we need to do the following steps. Excuse me. Figure out what alleles are in each of the parent's gametes. Okay. So in the F1 generation, we had capital T lowercase t for one parent, and the same for the other parent, capital T lowercase t. Right. Now, the second step is to create a table, the column for each possible allele for the male parent and each possible allele for the female parent. Okay. And then predict all possible fertilization outcomes by combining the letter from the row and the column in each box. So in the top right box, we have a capital T for one parent and a capital T for the other. So there's a T and a T there. After completing the Punnett square, we can see that 3 of offspring are predicted to have a capital T in their genotypes and therefore a smooth or round phenotype, while only 1 of the offspring are predicted to have a lowercase t and a lowercase t from 2 different gametes, and therefore have the phenotype of a wrinkled texture. And here's another example. So we want you to complete a Punnett square to predict the ratio of a cross between an F1 pea plant and a wrinkled pea plant. So let's go through the steps again. First, what are the genotypes of the peas that are being crossed in this scenario? Now you should know for an F1 pea that they're heterozygous. They came from 2 different true breeding plants, correct? One round, one wrinkled. So they have, when the 2 gametes combine to make a fertilized pea here, we're looking for capital T, lowercase t. Now you should also be able to infer what the wrinkled pea's genotype is. Why? A wrinkled pea cannot have a capital T or else it wouldn't be wrinkled. And so therefore, both of its zygotes are lowercase t. Now we don't have to write both lowercase t's because it's going to be the same on the top row as the bottom row. It's not going to affect our ratios because we'll get the same result twice. So we just write it down here once. Okay, so again, the F1P we know is a heterozygote with a capital T allele and a lowercase t allele. Wrinkled P cannot have a capital T allele and is therefore 2 lowercase t's. We only write 1 t down for the wrinkled P because the second row of lowercase t's will give the exact same result and therefore will have the same ratios. And so after filling out the boxes, our deductive reasoning of Mendel's principle of segregation would predict 50% round and 50% wrinkled seeds from this cross. And when Mendel performed this cross, he got 106 round and 102 wrinkled. So pretty darn close. Checkpoint 5. Which of the terms correctly describes each of the wrinkled parents in this cross? Let's review. So this is what we learned. Number 1, what did Mendel do in his first series of experiments? He selectively bred true breeding peas to create an F1 generation, and then counted the ratios, and then bred the F1s with each other to make an F2 generation. Number 2, based on the results of these experiments, what did he hypothesize about the way inheritance works? He hypothesized that inheritance was not a fluid mixture of parental genetics, but that each parent donated discrete alleles. Finally, that some alleles were dominant over other alleles, and their presence will define the phenotype of an organism. And number 3, what do we call Mendel's first hypothesis? And that is the principle of segregation. Mendel did not stop at crossing peas with one trait that was different between them. He also crossed peas that differed in 2 traits. For example, he mixed peas that were true breeding round and yellow with peas that were true breeding wrinkled and green. And he got a F1 generation that was smooth and yellow in the following ratios. So 100% of the F1 was smooth and yellow, but the F2, 9 out of 16 were yellow, 3 and smooth, 3 out of 16 were green and smooth, 3 out of 16 were yellow and wrinkled, and only 1 out of 16 was both green and wrinkled. While this result may seem unremarkable to us, what was interesting about it to Mendel was the fact that even though he started with yellow round peas and wrinkled green peas, in the F2 generation these traits were able to recombine with each other in ways that hadn't been seen in previous generations. For example, he saw wrinkled yellow peas in the F2 generation, when those traits had not been present in the same parent peas. The ratio for this 2-trait cross was again 9 yellow round, 3 green round, 3 yellow wrinkled, and 1 green wrinkled. And from this, Mendel's second hypothesis or principle addressed the question, why are the F2 ratios of a 2-trait cross always close to 9, 3, 3, 1? This is called Mendel's principle of independent assortment. The genes for any 2 traits, color, texture, height, etc., are passed down separately and not as a unit. The principle of independent assortment states that the inheritance of one trait has no effect on the inheritance of any other trait. Before we explain how this works in the context of Mendel's experiments, we need to establish the genotypes of the 2 parents. In this case, we're talking about 2 different traits, texture and color, and therefore we have 2 sets of alleles. We already learned that smooth texture is dominant, and the texture is signified with either a capital T or lowercase t, the lowercase t being wrinkled. And the yellow color is also dominant, and the color is signified with either a capital C or lowercase c. Smooth and yellow parent is a homozygous dominant for both. Wrinkled and green parents are homozygous recessive for both. So again, true breeding, they're going to be homozygous. Smooth and yellow are dominant. Now, every gamete produced by the smooth yellow parent would contain a chromosome containing the dominant allele for texture and also a chromosome carrying the dominant allele for color. Likewise, every gamete produced by the wrinkled green parent would contain a chromosome carrying the recessive allele for both traits. Therefore, no matter how the gametes from these 2 different parents combine, all offspring are going to end up with one copy of each allele for both of these traits. They will get a capital T and capital C for the smooth yellow parent, and a lowercase t and lowercase c from the wrinkled green parent. Because they have one copy of the dominant allele for both traits, the recessive allele is overridden, and all offspring present as round and yellow. Okay, so if alleles really do assort independently, then what does this tell us? This is where it starts to get tricky. It was easy to predict the different types of gametes that the parent plants would produce because they both only had one type of allele, dominant or recessive. And so there was only one option that could pass down in their gametes. The F1 generation, however, has a copy of each type of allele for each trait. So it can produce more than one type of gamete in meiosis. We can have capital C, capital T, lowercase c, capital T, capital C, lowercase t, or even lowercase c, lowercase t. All in all, there are 16 different ways that these gametes can combine, and you can model this much more easily in a Punnett square. A little messy here, the different ways that these F1 generation plants can combine. So let's follow the steps. Let's simplify things by doing a two-trait Punnett square. First, identify each gamete possibility for each plant and write them out. Along the top, we've written them out for one parent, and down the side, we have written them out for the other parent. Now we match the row and column to fill in each box. And now we are ready to analyze the ratios. All fertilized cells with a capital C will be yellow, and all with a capital T will be smooth, leaving us with 9 out of 16 offspring as yellow and smooth. Because green is recessive, we need both alleles to be lowercase c, and this means that only 3 out of 16 offspring are green and smooth. And because wrinkled is recessive, we need both alleles to be lowercase t, and this means that only 3 out of 16 offspring are yellow and wrinkled. And only 1 in 16 cells will be homozygous for green and wrinkled. It takes a gamete from both parents that are lowercase c and lowercase t to make this fertilized egg. And now we can see the ratios for the phenotypes of an F1 two-trait cross. Might take a couple of times of reviewing that process there before you get the hang of it. But once you get the hang of it, trust me, it gets a lot simpler after you practice a couple of times. So this is how we've arrived at this combination for the F2 generation. And you will need to be able to do this all on your own if given the genotypes of the parents. Let's predict the outcome of a cross between the parents shown at the left. Take a moment to pause the video and quiz yourself. Can you see if you can set up the steps all on your own? Okay, now let's go ahead and proceed and let's do it together. Start by determining what combinations of alleles would appear in the gametes of each parents. Here they are. Capital C, capital T, capital C, lowercase t, or lowercase c, capital T, or lowercase c, lowercase t. Now for the green wrinkled, because it's homozygous for color and texture, we're just going to get lowercase c and lowercase t. It's the only option through meiosis. Next, write out one set of gametes along the top for columns and the other down the bottom for the rows. And again, because this is homozygous, we don't need to write several rows for different CT combinations. It's not going to affect our ratios. So we get one row Now we're going to fill in the boxes. So in the first right box, the first left box, we're going to get a lowercase c, capital C, and a lowercase t, a capital T. And we just go through and fill out all of those boxes. And then finally identify the ratios of different phenotypes. So yellow smooth is going to be 25% for 1 of 4. Green smooth will be 25%. Yellow wrinkled will be 25%. And green wrinkled will be 25%. And here are Mendel's results, quite close to a 1 to 4 ratio for each. Okay, checkpoint 7. Complete a 2-trait cross between peas with the following genotypes: lowercase c, c, t, t, and uppercase c, lowercase c, and uppercase t, t. What fraction of the offspring do you predict will be both yellow and round? So that's the part I want you to answer on the checkpoint. Mendel was fortunate to be able to analyze the alleles he did. While Mendel's principles accurately describe the correct mechanism for genetic inheritance, they do not accurately predict inheritance patterns for most genes. Why is this? It is very rare for one trait to be dictated by only one gene. Usually, nature's a little bit more messy than that. It's also rare for each trait to have only 2 possible versions of the gene, right? There might be several. Think of eye color, for example. It's also rare for one allele to be completely dominant and the other completely recessive. And so here's an example of that. We have here traits for coat color in goats. There are 4 possible alleles. There's B sub D, which is an allele for a dark brown, and it's dominant over all other alleles. Then you have B sub I, which is light brown, and it might be recessive to B sub D, but it's dominant over the 2 lower alleles, B plus and lowercase b. B plus is black, but it's recessive to the 2 upper ones, B sub D and B sub I, but dominant over B. And B is red, and that coat color is recessive to all of the above. So there's one exception for multiple alleles. Another exception is incomplete dominance, whereby the dominant allele does not completely override the expression of the recessive allele. Heterozygotes express a phenotype that is between both alleles. An example, in snapdragon flowers, the allele for red color is incompletely dominant over the allele for white flower. Heterozygotes appear blended, so a capital R lowercase r genotype provides a pink colored flower. An example of incomplete dominance in humans is sickle cell. The allele for normal hemoglobin protein is incompletely dominant over the allele for sickle cell hemoglobin protein. So heterozygous individuals who have both the normal and the sickle cell, they will have some sickled red blood cells and some healthy red blood cells. So this is incomplete dominance over the other. Homozygous for sickle cell disease leads to all red blood cells having a sickle cell shape, and the condition can be fatal. Codominance is another exception. In the inheritance pattern known as codominance, 2 alleles are equally dominant, and another allele is recessive to both. For example, the allele for blood type A, I superscript A, is dominant with the allele for the blood type B, which is I superscript B. This is why a person can have an AB blood type. The allele for blood type O, lowercase I, is recessive to both. So you won't find a person that is AO or BO blood type. If the person is O homozygous, then their blood type will be O, but not if it's heterozygous. And that's because O is recessive to A and B is dominant over O. The next exception are polygenic traits, meaning many genes that coordinate for one phenotype. So these phenotypes require instructions from multiple genes. An example is human height, which is not indicated by one gene, but rather a great many genes working together. Pleiotropic genes provide instructions for multiple traits. An example is the gene located at 12q22 through 24.1. When mutated, this gene causes both mental retardation and loss of hair pigmentation. Genes can even interact with other genes, and this is called epistasis. This exception to the rule, epistasis, is defined as where one gene interferes with or masks the expression of another. An example is Labrador retrievers. One gene determines whether their coat expresses color. Within that gene, color expression, capital E, is dominant over non-expression, lowercase e. A second gene determines what color their coat expresses. Within that gene, black coat is capital B, is dominant over chocolate coat, lowercase b. Checkpoint 6. What are all of the possible genotypes that Ladybug the retriever could have for the traits of coat color expression and coat color? Sex-linked traits are dictated by genes that are carried on the X and Y chromosomes. Inheritance of these genes behaves differently because the presence of these chromosomes differ between males and females. Example is the gene for baldness, which is carried on the X chromosome. And remember that males have an X and a Y chromosome, while females have two X chromosomes. The allele for balding, lowercase b, is recessive to the allele for non-balding, capital B. Unlike females, males only have one copy of the chromosome, X chromosome, so they will express whatever allele it carries for balding, whereas females could have one on each, so it'll be a little bit different. We'll see how that looks. So unlike females, males don't have a second copy of an X chromosome. They are XY. Women could have a balding gene on one of their chromosomes, not exhibit balding, and that could potentially mask the effect of a recessive balding gene, lowercase b. Men, however, only have one X chromosome. So if that chromosome has an allele for baldness, they will exhibit that phenotype. So let's run a Punnett square on sex-linked traits. First, list all possible gametes for the parents. One set along the top for the columns and one on the side for the other. Here we have the maternal gametes along the top, both being X chromosomes, because the mother doesn't have a Y chromosome. Along the side, we have the paternal gametes. And remember, this allele is only found on the X chromosome, so we do not write anything down for the Y allele. In this example, the mother carries a balding allele, but is not bald because she has a dominant regular allele, capital B. The father has a balding phenotype. And filling in the boxes, we can predict that 50% of the children will have the balding phenotype and can further subcategorize it as one male, which is XY, having the balding phenotype, and one female, which is going to be two X's, two lowercase b's. And now, checkpoint 7. Hemophilia is a disease that prevents blood clotting. The gene that causes hemophilia is carried on the X chromosome. The allele for having hemophilia is X sub H. It is recessive to the allele for being healthy, X lowercase h, and it's recessive to the allele for being healthy, X capital H. Follow the steps outlined in the previous slides for sex-linked traits and perform a Punnett square to cross a woman who is a carrier of hemophilia, which means that she only has one copy, not two copies, with a man who is healthy. Okay, so that man does not have hemophilia. What fraction of their offspring do you expect to have hemophilia? And what is the sex of the offspring that are affected by the disease? Okay, that will do it for this lecture. Thank you so much for joining, and we'll see you in the next one.